Hello, today we're going to talk about photosynthesis. So we'll take a quick look at photosynthesis. So first we're going to start off with a little reminder about the carbon skeleton, and then we'll take a deeper dive into photosynthesis.
The carbon backbones of organic molecules may vary in length, as shown here by a carbon molecule with 2 carbons on the left and another molecule with 4 carbons. Carbon skeletons may have single bonds or double bonds, which can vary in location, resulting in molecules with different properties. Carbon skeletons may be unbranched or branched, and some carbon skeletons are arranged in rings. Such variation in carbon skeletons is one important source of the molecular complexity and diversity that characterize living matter.
So the carbon skeleton is kind of the foundation for all living things. And when we are doing photosynthesis, we are building those carbon skeletons from carbon dioxide. So we're taking inorganic carbon and we are making it into organic carbon. So when we think about photosynthesis, we kind of want to think about where in the plant does photosynthesis take place. So when we think about that, it's really the green parts of the plant that the photosynthesis is going to take place in. That's going to be primarily the leaves of the plant. So we kind of need to take a close look at the structure of the leaves. So the leaves are going to be green because they contain chlorophyll, and chlorophyll is the green pigment that is found inside the chloroplast. So we look at the leaf structure. So if we take a cross section of the leaf actually has these cells in it, and inside those cells are where the chloroplasts are located. There are other cells in the leaf as well. We've got cells on the top and the bottom. Those are kind of more structural and protective cells. And then the cells inside are going to be the ones that have the chloroplasts in them. And the chloroplasts are really the ones that are doing the photosynthesis. And in order to do photosynthesis, the plants will need carbon dioxide, and they also will be giving off oxygen. That carbon dioxide and oxygen exchange takes place typically through the underside of the leaf through these openings called stomata. So carbon dioxide will go in, oxygen will come out, so gas exchange will take place in there. The cells themselves will be doing cellular respiration and photosynthesis, so they are getting some carbon dioxide from cellular respiration, and they are providing some oxygen for cellular respiration as well. But there is usually more oxygen being given off and more carbon dioxide in demand than just the mitochondria. So there is a gas exchange through the stomata. And so the stomata is going to be those pores that gas exchange takes place through. And the chlorophyll is going to be in the membranes of the thylakoids. So the thylakoids, if we take a closer look at the chloroplast is a double membrane organelle. And then it also has these thylakoids inside, and those thylakoids look like stacks of coins, but they're really sacs, membrane-bound sacs that are kind of stacked on top of each other. And a stack of those thylakoids is called a granum, or grana. And the grana is that stack of thylakoids. The purpose of the thylakoids, the reason why they're stacked like that, is because it's going to increase the surface area of those enzymes that are involved in photosynthesis. So the more enzymes and the more chlorophyll we can fit into that membrane, the more photosynthesis is going to take place. So this is, again, increasing the surface area necessary to increase photosynthesis. So the grana are going to be the stacks of the thylakoid that are inside of the mitochondria. And then the chloroplasts are going to also contain the stroma, and that's going to be the dense interior fluid. That's going to be in the liquid that's in the space that's filling around the thylakoids. So the stroma is the space, and that's where the Calvin cycle is going to take place. And then the thylakoids are going to be stacked in stacks called granum, and that's where the light reactions are going to take place. And then, of course, we've got the double membrane structures, an outer membrane and inner membrane. So looking at photosynthesis, similar to cellular respiration, there are stages. There's 2 stages in photosynthesis. There are the light reactions, which is the first stage, the photo part. And then the Calvin cycle is the synthesis part. So in the light reactions, what's going to happen is that's where water is split. And as a result of the splitting of the water, oxygen is released as a byproduct. And then we also have the reduction of NAD plus to NADPH. NADPH is going to be an electron carrying molecule that will carry the electrons to the Calvin cycle to drive that reaction. And then it also generates ATP from NADPH by photophosphorylation. So ATP and NADPH are made in the light reactions, and those 2 molecules then move to the Calvin cycle, and that's going to be the energy that drives the Calvin cycle. And then in the Calvin cycle, what's happening is carbon fixation is taking place. The carbon dioxide from the atmosphere is being converted into organic molecules. And we'll take a look at each of those 2 processes in a little bit more detail. So this diagram kind of sums up the light reactions and the Calvin cycle. So we start with the light hitting the chlorophyll and the other pigments that are in the thylakoids. And then the water is split and oxygen is given off. And as a result of the light reaction, ATP and NADPH are made. Then the NADPH and the ATP are used to drive the Calvin cycle, where carbon fixation takes place. Carbon dioxide is then converted into organic molecules. That's a quick overview of those 2 processes, but we'll take a deeper dive into each of those. So starting with the light reactions, that again is going to convert the solar energy into chemical energy, and that conversion is going to make ATP and NADPH. So the chloroplasts are kind of the solar-powered chemical factory, and their thylakoids are going to transform that light energy into the chemical energy. So in order to understand how that happens, we need to take a quick look at the energy that's coming from the sun. So at the top here, it kind of shows all of the energy that comes from the sun. Only a very small portion of it, this little sliver here, is what's visible. So the visible light spectrum is what we can actually see with our eyes. And the wavelengths of light for visible light go from 380 nanometers up to 750 nanometers. So the blues and the purples are down low, and then the reds are and the oranges are higher up in the wavelengths of light. And the plants can only use some of that. They're not using all of the wavelengths of light. So when we kind of look at the pigments in the leaves that are absorbing the light, the plants are absorbing a lot of the light, but they're not absorbing the green wavelengths of light. So the wavelengths that they do absorb are going to be kind of the reds and the oranges, and then the blues and the purples. And they are absorbing it and then using it for photosynthesis. The reason why leaves look green is because they're not using the green wavelengths of light. And so that's what's being reflected back or transmitted through the chloroplasts. And so we see that because that's what's being reflected back and hitting our eyes, and it's making the leaves look green. So plants don't use the green wavelength of light, and that is why they look green. They're absorbing the other wavelengths of light except for the green. So when we think about why plants are green, they have a lot of chlorophyll in them, and the chlorophyll is absorbing all the wavelengths of light except for green. And so they're reflecting back that green wavelength of light, and then they look green. Some plants have different shades. So there are some plants that kind of look purplish. They are not absorbing the purple wavelengths of light, and they are reflecting that back. So what we see with our eyes is what is not being absorbed and what is being reflected back. And when we look at the chloroplasts, they don't just have chlorophyll in them. They have other pigments as well. There's chlorophyll A, there's chlorophyll B, there's carotenoids, and those are just some of the some of the pigments that we see in our leaves and in our plants. So they're each going to be absorbing a slightly different wavelength of light. So if you look at the chlorophyll A, it's going to be absorbing a lot around, it looks like about 450 wavelengths of light. And then it also absorbs some over here close to 700. Chlorophyll B is absorbing a little bit more of the blue wavelengths of light and a little bit more of the orange wavelengths of light. And the carotenoids kind of absorb a lot of the purples and the blues, but they're not absorbing any of the reds and the oranges. So the different pigments are going to absorb different wavelengths of light, but collectively, they're going to be absorbing those wavelengths of light and using those for photosynthesis. And so an action spectrum is really showing you the effectiveness of the absorbed wavelengths of light to drive the process of photosynthesis. So an action spectrum is really showing you kind of what energy is being used for photosynthesis. And then an absorption spectrum is showing you which wavelengths of light are being absorbed by those pigments. They're very similar and they kind of overlap a little bit, but they are a little bit different in telling us different pieces of information about those plants and those pigments. So when we think about why the leaves change color in the fall, they are going to be changing colors because they do have different pigments in them. But what happens is the green chlorophyll pigments, they're going to die off first. And they're going to leave behind the other pigments that are then reflecting back those wavelengths of light that make them look orange and yellow and red. So we're seeing those pigments because the green chlorophyll pigments have died off. And then eventually those other pigments will also die, and then the leaves turn brown and they fall off, and then the plant goes dormant for the winter. But the reason why we're seeing those greens and the yellows and the oranges and the reds is because the green pigments have died first. Okay, so let's take a closer look at the light reactions in photosynthesis. There are two photosystems involved in the light reactions. There's photosystem 2 and photosystem 1. But both of those are going to have a reaction center complex, and that's going to be surrounded by light harvesting complexes. So if we take a look at that, here in purple is our photosystem, and we're going to have these pigments in those photosystems, and those are going to be absorbing the wavelengths of light. So the photons are your wavelengths of light, and they are absorbing that energy, and they're transferring that energy to this set of chlorophyll molecules, which are then going to get electrons excited. And those excited electrons are going to go to a primary electron acceptor. That electron acceptor is then going to send those electrons down an electron transport chain. So it's those pigments that are absorbing the energy and transferring that energy to the electrons to get those excited so they can go down an electron transport chain. So the light harvesting complexes are going to transfer the energy to the reaction center, and then the primary electron acceptor in the reaction center is going to accept the excited electrons, and it will send that electron down the electron transport chain. So that's going to happen in both photosystems, but photosystem 2 happens first, and then photosystem 1 happens second. And the only reason why they're numbered that way backwards is because photosystem 1 was discovered before photosystem 2. And instead of swapping the numbers, they just kept it that way. And they just named the second one discovered second, photosystem 2. But photosystem 2 actually takes place first. And what will happen with that one is those electrons that get excited will go down an electron transport chain, and they're going to generate ATP. And photosystem 1 works best at 680 nanometers for their wavelength. And photosystem 2 is going to get electrons excited and send them down an electron transport chain. And then that's going to generate NADPH. And that works best at a wavelength of 700 nanometers. So if we take a quick look at each of those photosystems, the first photosystem again is photosystem 2. So the light is going to excite all of those pigments, and they're going to get an electron excited. Then, because that electron gets excited and goes to the primary electron acceptor, what happens is we have to replace that electron. And the way we replace that electron is we take the water and we split it. And of course, there are enzymes that are going to do that. The split water is going to then release oxygen as a byproduct, but the hydrogens get split and the electrons go to replace the electron that went down the electron transport chain to the primary acceptor and then down the electron transport chain. And then the hydrogen ions will be used to generate a hydrogen ion gradient. And we'll see that a little bit later. But the splitting of the water, the purpose of the splitting of the water is to replace the electrons that get excited and go down an electron transport chain. And so as the electrons go down that first electron transport chain, ATP synthesis is going to take place. Hydrogen ions will get pumped into the inner thylakoid space, and that's going to create a proton gradient similar to what we saw in cellular respiration. And then the hydrogen ions are going to diffuse back into the stroma, and that's going to generate ATP through chemosmosis. And we'll take a quick look at that a little bit later. But the electron transport electrons that were excited in photosystem 2, going to go down an electron transport chain and generate ATP. Then that electron from photosystem 2 goes to photosystem 1, where the light gets those pigments excited and gets that electron excited. And then that electron is going to go down another electron transport chain. And as it goes down the electron transport chain, then that electron is going to go to make NADPH. So the NADP plus is going to pick up the NAD is going to pick up the electrons and hydrogen ions and make NADPH. So this diagram here kind of sums up the light reactions. So you've got, again, light hitting the pigments, getting electrons excited. They're going to go down an electron transport chain to generate ATP. Water is split, so the electrons can be replaced. And then these electrons in photosystem 2, light hits those pigments, gets that electron excited, so it can go down another electron transport chain where NADPH is made. So the light reactions is converting that light energy into the chemical energy of ATP and NADPH. All of that is happening in the thylakoid membrane. And so, again, the more pigments and the more photosystems you can fit in the thylakoid membrane, the more photosynthesis you're doing. So if we look at how cellular respiration and photosynthesis are similar, there are obviously some similarities and there are some obvious differences between the two processes. But both of them are going to be making ATP, and they're making ATP through chemosmosis. So if we look at a diagram of the mitochondria, compared to the chloroplasts. Both of them are going to have membranes that involve enzymes that are doing the electron transport chain, and they're going to have the ATP synthase embedded in those inner membranes. So that's going to happen in the inner membrane of the mitochondria, but that's going to happen in the thylakoid membrane of the chloroplast. But both of them have electron transport chains. Both of them are creating a hydrogen ion gradient. So the electron transport chain, as the electrons move down the electron transport chain, hydrogen ions are pumped into the inner membrane space of the mitochondria and inside the thylakoid, that small space inside the thylakoid. And those teeny tiny spaces are going to build up the hydrogen ion concentration, and they're going to want to go back to the stroma or the matrix of the mitochondria to go from high concentration to low concentration. And as they do that, they're flowing through the ATP synthase enzyme. And as they flow through the ATP synthase enzyme, ADP, an inorganic phosphate, is converted into ATP. So they're both making ATP through chemosmosis. Just slightly different locations. One's in the inner membrane of the mitochondria and one is in the thylakoid membrane, but the process is the same. And so that's a similarity between the mitochondria and the chloroplasts. Okay, so when we look at the ATP and the NADPH, those are produced in the light reactions. They're going to be used to drive the Calvin cycle. And so this diagram here, again, sums up the light reactions, but this kind of shows you the thylakoid membrane. And so you've got photosystem 2 right here in purple. You've got photosystem 1 here in purple as well. The light is going to hit photosystem 1, get electrons excited. Water is split to replace those electrons. The electrons go down the electron transport chain. And as they do that, hydrogen ions are pumped into the inner thylakoid space. And then that's going to build up a high hydrogen ion concentration. And those are going to diffuse back into the stroma through the ATP synthase enzyme to generate ATP. In photosystem 1, light again hits the pigments, gets the electrons excited, goes down another electron transport chain, where it then ends up going to make NADPH. Both the NADPH and the ATP go to the Calvin cycle, which takes place in the stroma, and those two energy-rich molecules will drive the Calvin cycle. So we'll take a closer look now at the Calvin cycle. So the Calvin cycle, again, is going to use the ATP and the NADPH to convert carbon dioxide into sugar. So we'll see how it does that. What will happen is 3 molecules of carbon dioxide will go into the Calvin cycle, and they will get converted into a molecule called glyceraldehyde 3-phosphate, also known as G3P. So photosynthesis doesn't exactly make glucose. It makes G3P, which is then used to make glucose. So the G3P is what comes out. Carbon dioxide is what goes in, and carbon fixation is the process of converting carbon dioxide into G3P. So the Calvin cycle has 3 phases, but we're not really going to get into those 3 phases. But there's a carbon fixation phase, a reduction phase, and a regeneration of the CO2 acceptor phase. But that's not really that important. Okay, so looking at the Calvin cycle, we've got the input of carbon dioxide. So 3 molecules of carbon dioxide will come into the carbon fixation phase, and it goes through a series of steps where it uses ATP and NADPH, and then G3P comes out. There's other molecules that will get recycled, and again, that's going to use ATP in the regeneration phase. But it is a cycle because those molecules are reused over and over again. But the carbon dioxide comes in, G3P comes out. The G3P can be used to make glucose and other organic compounds. And that is carbon fixation. And the ATP and the NADPH from the light reactions is what's driving that process. Okay, so this diagram kind of sums up the whole photosynthesis. You definitely want to take some time to study and analyze this picture. So again, the light reactions are going to be driven by the light from the sun. So solar energy is going to come in. It's going to drive the photosystem 2 and photosystem 1. Photosystem 2 is going to generate ATP. Photosystem 1 is going to generate NADPH. Water is split to replace those electrons, and oxygen is a byproduct. And then the ATP and the NADPH are used to drive the Calvin cycle, where carbon fixation takes place, and the carbon dioxide is then converted into G3P, which is then used to make other sugars. And so that kind of sums up the process of photosynthesis. And this diagram is just another diagram summing up photosystem 1 and photosystem 2 and those two electron transport chains. And then this is just another summary of the Calvin cycle. Carbon fixation takes place where carbon dioxide is converted into G3P. So it actually takes 2 cycles of the Calvin cycle to make 1 glucose. So you'd need 6 carbon dioxides to make 1 glucose. All right, so let's take a quick look at a little video that sums all of this up.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores, and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The 2 green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the 2 photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced 2 energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With 3 simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
Okay, so that's a nice summary of photosynthesis, each of the steps, and I like the animation kind of shows you visually how that whole process is taking place. The one quick tip for this unit is definitely take the time to study the diagrams that are in the unit. So there is the diagram for cellular respiration. So definitely take some time to understand and be able to explain this diagram. And then of course the diagram of photosynthesis, take the time to study and analyze this diagram as well. If you can explain the diagrams, then you can really understand photosynthesis and cellular respiration. And then this diagram kind of shows you how this unit is tied to other units. So you've got some of that movement across the cell membrane, you've got energy transformations in the cell. So, and that ties all of those units together. We'll also see how it ties to the flow of genetic information a little bit later on and making proteins and all of that stuff. But this is a nice diagram to kind of sum up that connection. And you want to take some time to also fill in the blank photosynthesis diagram to quiz yourself, see how you are doing remembering each of the different steps. So it's a good idea to take the blank diagram and see if you can fill in that blank diagram and quiz yourself. So let's take a quick look at that. And if you have any questions, please let me know.